Good afternoon, students. Today, first we will discuss the topic characteristics of genetic material. So, what are the criteria which should be fulfilled by a material or a substance to become the hereditary material or the genetic material? The first point is it should be able to generate its own replica by the process called replication. It should produce its clone. Next, it should be chemically and structurally stable. It should not be destroyed by any way. Next, it should undergo slow and gradual changes called mutation. So we know that mutation is the root of all evolution. So hence, the genetic material should be able to mutate slowly to form more evolved organisms. It should have the capability to express itself in the form of Mendelian characters. If the particular material or compound have these following characteristics, then they are suitable, or you can say they are rightly be known as genetic material. Next, DNA replication or replication of DNA. The first point, which is the criteria of being genetic material, that it should be able to replicate. So what is DNA replication? In molecular biology, DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. This is the original molecule, exact copy is produced. So this is DNA replication. So why these DNA replication should take place? DNA replication needs to occur because existing cells, this is the existing cell, divide to produce new cells. Each cell needs a full instruction manual to operate properly. So, the DNA needs to be copied before cell division so that each new cell will receive full set of instructions. So, what is the mechanism behind the replication of DNA? How DNA replication takes place? So, regarding the mechanism of DNA replication, several hypotheses were put forward by many scientists. But the theory proposed by Watson and Crick is now highly accepted. They proposed the semi-conservative theory of DNA replication. According to their semi-conservative method of DNA replication, the double-stranded DNA molecule separate, as you can see here, separate. Each separated strands act as a template strand. Each template strand can synthesize a new complementary DNA strand. The complementary DNA strands then binds with their respective parental strands. Thereby, two molecules of DNA are formed, each having one parental strand and one daughter strand. So this is the semi-conservative DNA replication proposed by Watson and Crick. The semi-conservative DNA replication proposed by Watson and Crick was actually proved experimentally by two scientists, Matthew Meselson and Franklin Starr. They proved that DNA replication actually follows the semi-conservative method. So, for their experiment, they took Escherichia coli. They cultured Escherichia coli in a confined condition, in a suitable medium. At first, they cultured the Escherichia coli in a medium which contained heavy nitrogen. This is the normal nitrogen which is present in our DNA. This is the heavy nitrogen. In this medium, they cultured E. coli till the bacterial DNA becomes completely labeled with the heavy isotope nitrogen. Normally, the DNA in its structure possesses the normal nitrogen. Now, if we consider the position of these heavy nitrogen and the normal nitrogen in a centrifuge tube, the heavy nitrogen occupies the bottom of the centrifuge tube, and the normal nitrogen, it occupies the surface. Okay. Next, the heavy isotope nitrogen DNA can be separated from normal DNA by density gradient centrifugation. Just now I told you here. The labeled bacteria were then shifted to a fresh medium which contain normal nitrogen. Samples were taken after regular intervals, and the DNA extract was subjected to centrifugation. To measure their densities. It was observed that the DNA of first generation was of hybrid or intermediate density. That is, they occupied the middle part. Not the surface layer, not the bottom, but the middle part of the centrifuge. The second generation of bacteria consists of, when subjected to centrifuge, consisted of equal amount of light DNA and hybrid DNA. Thus, their experiment proved the semi-conservative nature of DNA replication. Thank you.